We want to take a look at the most common fossil that we find in the geological record that is indicative, or at least possibly indicative, of microbial life. We've already discussed that microbes make up 60% of what's living on planet Earth, and therefore you'd reasonably want to then find some kind of a fossil record of microbes to help understand the history of that organism that dominates our planet.
So what do we mean by microbial life? Well, the microbial life that actually was occurring during the Archean was cyanobacteria. So a lot of people get cyanobacteria confused because it is green and it photosynthesizes, which is not necessarily indicative of just a plant. Bacteria actually were one of the first organisms to photosynthesize before plants even appeared on the earth. So cyanobacteria are actually bacteria that have cells, but they lack some of the characteristics of eukaryotes.
So the primary type of rock that houses this paleontological fossil evidence of microbes is called a stromatolite. And the stromatolite is actually a highly controversial term. The reason it's controversial is not because of what it describes, but because of what it might indicate. So the word stromatolite is actually a finely layered sedimentary rock, layers of rock that are on the scale of tens of microns to hundreds of microns to millimeters. So very finely laminated layers of rock that have been accumulated through geological time, with the oldest at the bottom and the youngest at the top.
So stroma in Greek means layered. Light or lithos in Greek also means rock. So stromatolite is just layered rock. Very simple, very easy. And these layers are very, very thin. And so how I usually try to understand stromatolites are that most stromatolites, not all, but most stromatolites are actually produced by some kind of biological indicator or force, such as cyanobacteria.
Now, we know that in the modern oceans, microbes can grow on the seafloor, and as the microbes grow, they make a sticky, gooey mat that sticks together the cells and protects them from the rest of the marine environment. And because it's sticky, it actually allows sediments, small grains of sand and clay, to stick and accumulate. So as the microbes are growing on the seafloor in some places, the sand grains and the clays, they accumulate and they make layer upon layer. And these layers, again, are millimeters or smaller.
So the reason why cyanobacteria actually grow through the sediment and create a whole another layer is that cyanobacteria are photosynthesizing, so they require sunlight. So if you have sediment on top of you, you can't get any sunlight at all. So they have to actually grow through it and create a whole another layer. And eventually more sediment will come on top of them and they'll have to grow through that again. So that's why you get multiple layers of microbial mat and sediment, microbial mat and sediment.
So those occurrences in the modern day ocean where we know there are microbes growing and accumulating these kind of sediment layers, those stromatolites are known to be of biological origin, and therefore they are a living preserved record of microbial life. Now that's all fine and good for the modern, but one of the problems is when we go to the ancient geological record, we find immense deposits of stromatolites. We find stromatolites that are literally kilometers in thickness. And these were growing on ancient seafloors or sometimes in caves or even in hot springs. So stromatolite deposits, finely layered sedimentary deposits, are very, very abundant. But the million-dollar question is, which of those stromatolite deposits are microbial in origin, and which of those can actually be formed by non-biological processes? So the idea of having very thin layers of sediment that are laid down one after another through geological history, as I mentioned, it can happen when microbes are present, and the microbial biofilms, the mats, stick together the sediments. But we also have very good evidence from experiments in the laboratory and also modern natural settings where we can make these finely layered sediments and there were no microbes around whatsoever. There are details in the structure of the stromatolites. Things like, well, sometimes the stromatolites make a bulbous, almost pillow-shaped deposit. And parts of the bulb are anti-gravity, if you will. And the thinking there is that, well, you can only stick sediments together to make a layer and have those be anti-gravity, sticking against themselves, if that stickiness is driven by some kind of microbial mat. But what we see is that there can be changes in temperature and viscosity and density of the water, and these factors combined with a myriad of chemical changes that can actually make a non-biological stromatolite. So stromatolites are critically important to us. We know that some large proportion of them are microbial in origin, but understanding which are microbial and which are not takes an extremely intensive and long amount of work on any one geological deposit to make that inference. As NASA lands spacecraft on other planets, like the Curiosity is doing now, we're hoping that one of the types of rocks that the Curiosity will collect is a stromatolite, because it is possible that those might be microbial mat deposits from ancient life that occurred on Mars. But careful evaluation will have to be done to be able to prove or disprove the concept of whether those finely layered sedimentary structures are biological in origin or non-biological in origin. So understanding the fossil record in this context, this ability to go into the ancient rocks, either on another planet or on Earth, and decipher whether or not they are the product of biological activity or non-biological activity is kind of the fundamental boilerplate of us exploring the ancient life that occurred on Earth, as well as the ancient life that potentially has occurred on other planets.